Okay, so this is the third lecture of the week, chapter 17. These chapters are quite abbreviated, as you see. Again, you're only responsible for knowing what's in the slides, not everything in the book. So just focus on what's in the slide and focus on that portion of the book for the exam coming up. This links with our immune system. So we covered the innate immune system, and then we went to the adaptive immune system. And now we're going to look at how vaccinations work with the immune system. So we're going to look at the key concepts of vaccination and immunological terms coming up. So looking at active immunization, we spoke about this a little bit earlier in our last lecture, chapter 16. We have the options of injecting an antigen, which will stimulate the immune system. So this could include an attenuated microorganism. This could contain a killed microorganism or just a piece of an antigen where the epitope will elicit an immune response. So this is what our vaccines are. They're active immunization. So they're eliciting an immune response. Passive immunity, on the other hand, does not elicit an immune response, but provides protection. So essentially, you're giving antibodies to someone. So we can purify antibodies and administer antibodies that can help agglutinate or find cells or microorganisms within the body that are infected. And that can help deal with an infection. So we can have passive immunization through breastfeeding. So antibodies are passed from mother to child to protect the young because their immune system is not fully developed at that point. So active immune responses create memory, meaning that you'll be able to identify a foreign invader a second time around. Passive immunity, on the other time, does not have memory. It's only antibodies that are given to you, not antibodies that you generated yourself. Okay, so looking at what an effective vaccine is, we probably know a little bit more about this from going through the pandemic. We know that we don't want the vaccine to harm the human. Vaccine should be able to stimulate B cells and T cells so that we can have memory of it. So the main purpose is to get this immunological memory of the foreign invader so that if we do get exposed to it in a natural environment, our immune system will recognize the foreign invader immediately and be able to mount that secondary response that we talked about in the previous chapter, chapter 16. The other thing is we want to have not too many boosters. So we don't want to have to keep boosting our immune system. A good vaccine requires only a booster or two. The other thing we want to know is that it doesn't only cover you against viruses or bacteria in a lab setting. It actually protects you against natural pathogens. So the vaccines that we generate annually, maybe the flu vaccine, we want to make sure that it actually works against a natural pathogen. So those are the projected vaccines. Okay. So again, we have vaccines that can be killed. So we have whole cells that are inactivated, dead organisms. So these get a large immune response. We also have live or attenuated weakened viruses. A lot of times these are mutated so that so that their growth is essentially diminished within the cells. These can create some of the largest immune responses because they can actually start an infection cycle. While they can't cause infection, they can start some of the natural infection cycles. Versus if we're looking at purified components like an antigen itself that's being injected, we're not going to see the entire infection cycle. So our immune system has a little bit less to respond to, but these are probably some of the most common vaccines. And then more what we're seeing today is our DNA or RNA vaccines. We see this as not commercially available. Well, since the pandemic, that has changed, and the book is not updated since then, but that has changed. We have our RNA vaccines for COVID. Okay. Let's look at herd immunity and see how herd immunity works. So herd immunity is extremely important for protecting our population and specifically for protecting our immunocompromised portion of the population. So this could include children, this could include seniors, this could include people with immunodeficiency disorders, people with organ transplants. All of these things are reasons why we want herd immunity to protect the population. So herd immunity normally comes about when we have 3/4 of the population that's immune. And this table that this image that we're looking at right here is really going to help us understand kind of what's going on. So let's look at the first example here. Here we see that no one is immunized. So in the situation, we have healthy, unimmunized in blue, and then we see our sick, non-immunized people. So we have two of them within the population. What will happen is that this will spread quickly throughout the population, and rapidly, we're going to see that more people are sick. So again, this is without any immunity, without any vaccination within the population. Now let's look at this second example, example B. In yellow, we see these are people that are healthy and immunized, and then in blue, people not immunized. What we see is that while we have spread of the contagion, it doesn't spread as quickly through the population as what we saw before in example A. So the red ones are the infected ones. Down here, we see less because we have some people that are preventing the virus from spreading from person to person because they are immunized. Now in C, when we see a very high percent of immunization, again, we want about 3/4 of the population immunized, depending on the disease of interest. And we have two infected individuals. We had two here, we had two here. We see the spread of the red infected individuals is diminished because we have so many people that are immunized, and they end up protecting the non-immunized individuals. So non-immunized individuals, it's not always people who may be anti-vaxxers. There's some people who can be allergic to vaccines, who cannot get a vaccine. There might be an age restriction on a vaccine. There might be a financial constriction. There might be an equitable issue of the vaccine availability. So just because someone isn't immunized doesn't mean necessarily they don't want to be immunized. And that's why herd immunity goes and protects a good percent of the population that's not immunized. So again, example A, we have two people not immunized that are sick, contaminating almost the entire population. In example B, in yellow, we have some people immunized. It slows the spread of the contagious disease. In example C, again, two people not immunized, we see that it still spreads, but it's pretty much contained because we have about 3/4 of the population that are immunized. And that's why herd immunity can protect us as a population. Okay, let's look at opposition to vaccines. So there's the data surrounding vaccines are indisputable. There's no link to autism or diabetes in any of the common papers that we've seen. And let's go ahead and see how they've worked. Here we see measles progression, number of cases in the thousands here. Here we can see measles vaccination programs begin for immunization, and we see the measles cases drop. So in 1968, a drastic drop all the way up here to 2008. And following that in 2008, we see a spike in number of cases. Why do we think we see a spike in number of cases here? I'll give you a second to think about that. Vaccinations came out, cases dropped. 2008, all of a sudden we see a spike in measles, a measles outbreak. That's because people stopped vaccinating their children, because people stopped believing that vaccines were beneficial to humans and that there was a potential link to autism and diabetes, which goes against the scientific evidence that we've seen. So from this graph, we can see that vaccines save lives, millions of lives. We see the expected life of people have been extended because of vaccines. Children are living to longer ages because of vaccines. So similar to antibiotics in their effect on the population, we've increased life expectancy with them. Okay. Let's look at diagnostic tools for immunology. So diagnostic tools are a way we can assess maybe vaccines. And this is specifically what I worked in is diagnostics. So I worked in a oncology diagnostics laboratory at Roche Molecular Systems and a small biotech startup in Los Angeles. And what we worked on specifically was these two factors, specifically specificity and sensitivity. When we're looking at like blood or something in a serum, contents in a serum, we could look at antibodies or antigens in a serum. We can look at the test specificity and sensitivity to it. So specificity is how accurate the test is. We want to make sure that it measures the selection of an antibody towards its antigen. So we want that lock and key fit. We don't want antibodies that are grabbing all different types of antigens and alerting the immune system. So in diagnostic tests, if we have a test that's going to test for COVID, we want to make sure that we have specificity. We don't want to pick up other SARS-related viruses. So we need specificity first in order to work on an assay. And we also need sensitivity. If you remember, looking at the types of assays that we've had for COVID, there was a PCR-based assay that was more accurate. Then there was also the take-home test assays, which were less accurate. And that had to do a lot with the sensitivity itself. So sensitivity is how low of antibody or antigen that can be detected in a sample in order for the test to turn positive. So if you did an at-home COVID test, the sensitivity of those are not great. So you could have a COVID infection, but not a high enough viral load that your body, that the test would actually pick it up. So whether it was picking up antibody or antigen, the test wasn't able to pick it up. So you would have to wait a couple more days, test again to see if it became positive. But if you did a PCR-based test, PCR tests were, again, more accurate, more sensitive, so they could pick up a lower viral load. They could detect an infection earlier on. So sensitivity and specificity are important when designing assays to pick up microorganisms. So again, specificity has to do with actually identifying the right microorganism, the right strain of that microorganism, and sensitivity has to deal with how much of the microorganism or antibody has to be present in order for the test to pick it up. So a test can pick up an antigen that would be a direct correlation with a microorganism. So if we're picking up the surface spike protein from the COVID virus, that would be the antigen, the specific epitopes that the test is looking for. Or we could look at antibodies. We could look at your response. If you have already been infected with COVID, you would have generated antibodies for it. So we can look for either of those, antibody or antigen. Looking for antigen is showing an active infection. Looking for antibodies is showing a past infection. Okay, and that is it for this short lecture.